Take a look around your kitchen. You likely have a bread loaf, some cheese in the fridge, and wine on the shelf. These common pantry items share a surprising connection with advanced lab-grown medicines and even the science of genetic cloning. That connection is biotechnology. A scientist named Karl Ereky coined the term in 1917 to describe a simple idea: using living systems and biological processes to create useful products for people. While it sounds like a futuristic invention, this is actually the oldest technology we own. It has been a part of human life for thousands of years. To see the start of the story, we have to travel back about 10,000 years to the Paleolithic era. This is when early humans first began acting as biotechnologists. Instead of just hunting whatever they found, they started practicing selective breeding. They carefully chose the best wild sheep and cattle to live near them, slowly domesticating animals with the traits they needed most. They applied this same logic to their food. Ancient Egyptians and Romans discovered that by letting crushed grapes or dough sit out, wild yeast from the air would transform them into wine and bread. But there was a catch. These early farmers were acting on observation rather than evidence. They knew the dough would rise if they waited, but they had no way of knowing what was actually making it happen. For millennia, human survival relied on a partnership with an invisible workforce of microbes that we couldn't even see. That invisible world finally came into focus in the 1800s. As microscopes became more powerful, we stopped guessing and started seeing. This is Louis Pasteur. He was the first to prove that fermentation wasn't a random or magical event. He showed that living, breathing microbes, like yeast, are the tiny workers responsible for the change. They grow by digesting sugars, and the substances they leave behind are what turn grape juice into wine or dough into bread. This realization was the turning point. Biotechnology was no longer just a traditional art passed down through generations. It was now a precise science we could study and control. By the 1900s, global warfare created massive crises that required us to take these microscopic processes out of the kitchen and move them into massive factories. During World War I, Germany needed a steady supply of glycerol, a chemical used to manufacture powerful explosives. To get it, they learned how to use industrial-scale fermentation to brew the chemical in giant vats. Then, in World War II, the focus shifted to saving lives. Thousands of soldiers were dying from infections on the battlefield, creating a desperate need for a new kind of medicine. This led to the invention of the modern bioreactor, controlled tanks that allowed us to mass-produce the world's first antibiotic, penicillin, with extreme precision. By the middle of the century, we had mastered the art of farming microbes for the useful substances they naturally make, but we were about to learn how to change what they make entirely. The limit of those early factories was that they could only produce what a microbe was naturally programmed to create. We were still bound by the laws of nature. That changed with recombinant DNA technology. It works like a biological cut and paste tool, allowing scientists to take a specific genetic instruction from one organism, splice it into the DNA of another, and insert that custom blueprint into a host cell, like a bacteria. This gives us the power to write the software of life ourselves. We can now give a cell a new set of instructions, turning it into a microscopic factory that builds exactly what we need. This is how we produce human insulin today. By placing the human gene into bacteria, we can mass-produce life-saving medicine for millions of people around the world. This same logic of genetic control eventually led to milestones in animal science, including the successful cloning of an adult mammal, Dolly the sheep. So when you look at that loaf of bread or block of cheese on your counter, you aren't just looking at food. You are looking at the early chapters of a 10,000-year story. We have moved from simply watching nature work to learning how to speak its language, using the power of biology to build a healthier future.